Okay, well, yeah, I'm really glad to be here and to be back after I've stopped in once or twice since I graduated with my PhD 8 years ago, but it's been a while since the last time, so it's nice to be back and to see a lot of faces that I recognize. So Jim Curley invited me last fall to come and join the seminar series, and specifically to talk about some work I did during my postdoc in Iowa related to cover cropping. So I'm going to start with that after I just have to start first with this brief little aside. So I was digging around in my yard recently and found a bunch of silver spoons, and it ignited really a new interest for me, which is to make jewelry out of spoons. And so I just wanted to spend a few minutes to show you some of the things that you can do. And so a lot of these older spoons, they actually have like real silver in them. And with just minimal processing, you can convert kind of junky things into treasures. And so I just have 9 or 10 slides about silver spoons. So.
Are you selling these afterward?
No, that's my April Fool's joke. My wife is really big into April Fools, and she told me I was not allowed to do this presentation unless I tried to make some kind of a joke. So that's my attempt. All right, so I think of myself as a microbiome scientist or a microbial ecologist who's interested in thinking about microbes at the community scale. And I think probably everybody by this point has become familiar with the term microbiome. Is anybody unfamiliar or uncertain of its meaning? I find it unfortunate that we've adopted this pronunciation of microbiome, and I think the meaning is more clear if we would say it as microbiome, which links it more clearly to genome and proteome and metabolome. So the concept of, or that suffix of ome just means the entirety of something. So we're trying to look at all the microbes together rather than only a small subset like a single species or something. Trying to be more comprehensive in how we look at microbial communities. So I'm going to tell two mostly unrelated stories, but the first about cover crops. So I was hired by an agronomist who had a hunch that there was a disease problem in some of his cover cropping trials to do a postdoc to kind of lay out this, test this. Is it really a disease thing or what's going on? So cover cropping is primarily motivated by conservation concerns, soil and water quality, aiming to prevent this type of travesty from happening in agricultural fields. So instead of having bare soil that's easily eroded and degraded, we'd have some vegetative cover in place over as much of the year as possible to protect that soil. So this is one of our field trials. So you can see what this would look like in practice. Cover cropping often accompanies no-till management. If you're going to till, you basically don't have enough time to cover crop as well. And it's kind of part of the same philosophy about how to maintain soil quality and things. So here's corn growing in a long-term experimental field that's had cover crops. And all the brown in the interrows is cereal rye, which was planted the previous fall, grew in the fall, overwintered, grew more in the spring, and then was actively killed with an herbicide ahead of corn planting. So that's what the practice looks like. You can kind of tell that the next experimental block here has plants that are taller and greener, and that is one of the effects of cover cropping, is this delayed and somewhat chlorotic type of growth. So we know that there are things about the practice that make it harder for corn, and some of those are just physiological, it's about soil temperature and whatever. But the question was, is it also a disease thing? You can't see in this picture effects on anything like stand density or whatever, but those effects also manifest themselves sometimes. So this is the same view later in the summer, and you can see, hopefully, that all of that cover crop biomass has disappeared. And again, it's a never-tilled system, so it's just the biology of the soil is rich enough to process all of that material, and it's basically gone. Whereas I can see, and maybe you can still see some of the corn residue from 2 years previously. So the bigger corn stalks and stuff have a much longer longevity in the field compared to these cover crop residues, which disappear relatively quickly. So I'm not going to talk much about methods, but microbiome profiling mostly is a form of, or relies on amplicon sequencing. So generate more copies of some marker gene by PCR and sequence many millions of those copies and infer from those snippets of DNA sequence information something about the organisms that are present in that environment. So in this cover cropping experiment, we were interested in what was the microbiology happening in these plants as they were dying, the cereal rye cover crops. So they're being deliberately sprayed with an herbicide to kill them, and then very shortly after, or sometimes even before that, the corn is being planted right into that residue. So we started out looking in the roots of the rye with these type of microbiome methods to ask what's happening to the microbiology as those tissues die and over time as they begin to decompose. And so one of the primary management decisions that farmers can make if they're cover cropping is when to kill the cover crop. And so we also incorporated different kill dates and sampled repeatedly over time. So the first, I'm just going to make a couple of summary points about this data set without belaboring kind of how we process it or anything. The first thing is that this type of microbiome profile is going to introduce you to many, many organisms that you know nothing about. So here's the first example. Pretty common in the roots of these cover crops were DNA sequences that looked like they belonged to something called Legina radicucola, which I had never heard of before. And they followed kind of an interesting trend pattern where so DPS here is days post spraying. So this organism was more abundant in the roots early on and declined precipitously over just a few weeks. So looking up some information about it in the literature, it's an obligate parasite. So it's a biotrophic organism, which you would never see in a petri dish, or if you tried to culture organisms out of these roots, it would never show up anywhere in your survey. So that's the first point, is microbiome profiles let us see things that we can't see through culture-based approaches yet. And the pattern of loss over time fits with its obligate biotrophy, that when the host dies, then this organism also is quickly displaced from those roots. Interesting tidbit of biology that I didn't expect. Was there a question there? Okay, and then another point is just the throughput that you can do with microbiome profiling. So it's easy to process dozens to hundreds of samples and get a snapshot across the community of which organisms are present there. And doing that generates lots of new hypotheses. So here's just an example point from this data set. So in the roots of these cover crops, across this one experimental field that was something like 300 or 400 meters in extent, there's almost this perfect switching of the dominant Pythium species across that field, where there's sequences that look like they belong to Pythium volutum at one end of the field and they drop off by the other end and are replaced by this other Pythium sequence that doesn't look quite like anything that's been described. So in this particular field, there happens to be an elevation gradient that matches this switch in dominant species almost perfectly. And it's not much of an elevation gradient. If you just look at it, you could say, yeah, it's like, there's a slight slope there, but it's not very obvious. Less than 4 meters across this rectangle. So is it the elevation difference that explains why one taxon is dominant over another? Or is it something that's not captured yet in this information? But you could explore a lot of hypotheses out of this type of a data set that you maybe wouldn't have generated in any other way. So those two dominant Pythium species, the first one, as I mentioned, doesn't seem to fit anything that's been described, but we were able to go, after seeing that, we went back to the field and could isolate this organism out of the roots of these rye plants. So first seeing them as an amplicon sequence in a microbiome profile, we were able then to return to the site and get the actual organism in hand. And the second one was up here, Pythium volutum, and that's one that, so I worked closely with Alison Robertson at Iowa State University on this, and does a lot of field research with Pythium, and that name just didn't even ring any bells for her, and yet it was the dominant species that appeared among the Pythium in these roots. So going through the literature, there are a few references to it from turf grass and from wheat in the Pacific Northwest. And interestingly, all of these references comment on how hard it is to get this thing to grow on the typical selective medium that's used to isolate Pythium. So that was interesting, and I thought, again, I should go back to the site and try to get this organism in hand and validate that what I see in that sequence data is something that's actually there. So there's one strain of this species in the American Type Culture Collection, and working with that strain, I could show that it was extremely sensitive to PCNB, which is typically used as a selective agent in Pythium isolation media. And it's also, to a lesser extent, sensitive to some of the other antibiotics that are typically in there. So by omitting PCNB and reducing the concentration of other antibiotics and isolating more strains, we were able to get our hands on this organism that we first saw as just a DNA sequence in a microbiome profile. So that little bit of microbiome profiling in the cover crops is kind of like the whole microbiome component of this work, and then the rest of it is follow-up. But I think that's important because I don't really think microbiome profiles can be the end product of a research program. They're more of hypothesis generation, and you have to do some kind of specific follow-up testing most of the time. So when we had isolated out of those rye roots, we were getting a variety of Pythium species. Only with special techniques that I just described did we get what appeared to be the dominant species based on the DNA. And both of those belong to sort of a clade within Pythium, which is called the B clade, or Pythium clade B. So then we could narrow in on either a species of interest like Pythium volutum or a broader group like the B clade of Pythium that our microbiome profile suggested were important, and do more targeted tests on those organisms. So here now we switch from looking in the cover crop roots to looking in the roots of the corn crop that's planted into that residue. And with a qPCR assay for Pythium volutum, we can show that corn that follows a fallow treatment has less of this pathogen than corn that follows a cover crop. And with isolates in hand, we can do pathogenicity assays and demonstrate that these organisms not only can move from rye roots into corn roots, but that they are true pathogens and capable of causing disease. So most notably, they kill the radicle sometimes very soon after emergence, and sometimes a little bit later, but the radical tip necrosis is going to be detrimental to corn seedling performance early on and things like that. So they are legitimate pathogens of corn, which had not been described before. Okay, so then it was interesting that they were both of those dominant Pythium were in the B clade, because we isolated a lot of other Pythiums from primarily clade F, but also other parts of the Pythium phylogeny. So does cover cropping, elevate Pythium infection risk in corn uniformly, or is it specific to these taxa that do very well in rye roots, or those type of questions? So again, with quantitative PCR now at the clade level specificity, so here's clade B and here's clade F, and we're seeing infection in corn roots following fallow in the first 2 bars, or following a rye cover crop in the next 2 bars. So you can see that the corn infection rate by the B clade responds dramatically to cover cropping treatment, whereas the corn infection rate by these other Pythium species is non-responsive to the cover cropping treatment. So we're building evidence that a specific pathogen is responsible for this corn performance issue following rye cover crops. So then if you want to get to some kind of management recommendation from that, I mentioned already one of the primary decisions a producer can make is when to kill their cover crop. So that's something we tested experimentally. So now days before planting or days after planting. So this refers to how far in advance of corn planting was the rye cover crop killed, or was the herbicide applied. And here we have the infection rate in the corn by this Pythium clade beet. So if you plant your corn and then kill your cover crop later, 2 days later, you get this high level of infection. But as you kill your cover crop increasingly early, that drops off. So this is a log scale, so it's a bigger jump than it looks like. But even several weeks lead time on killing your cover crop doesn't bring that infection rate down to what it is in the fallow control. So why cereal rye? Maybe that's a bad cover crop. I mean, it's a grass like corn's a grass, and you could expect that they would share pathogens, and so we're kind of documenting the obvious to some extent. The problem is that cereal rye is really the most reliable cover crop for the conservation benefits that we're trying to get out of it. In terms of winter hardiness, it will almost always survive the winter. In terms of biomass production, it grows enough to be meaningful, which a lot of other cover crop candidates just don't produce enough biomass to matter. So that's why rye is used so much, but we can show by contrasting rye with some other potential cover crop species that this risk of elevated infection in corn by this particular clade of Pythium is really specific to rye and is not shared by alternative cover crop species. So it would be great if we had a good performing cover crop that was not so closely related to corn and might not share as many pathogens as corn. That would be fabulous. Okay, so that's kind of the cover crop story. Just one more picture of what it can look like, and I think it's hopefully obvious kind of why that would be a desirable state on the right and somewhat less desirable on the left. This is a corn silage production field, so very little residue is left on the field. But what I want to highlight here with this picture is just another management option, which is that we can precisely locate where we place something like cover crops relative to the crop row and maybe reduce our disease risk in that way. So this is showing placement relative to the old, the previous crop, but you could do a similar thing where you would avoid planting your cover crop in the space where your next corn row is going to go, but plant all the inter-rows and give your corn seedlings a little bit more space from those pathogens that are hosted by the dying cover crop roots so that they can establish before they need to mount their defenses and whatever. So I'm going to switch to a pretty different story. Any discussion about cover crops and disease risk or questions for me on that? Debbie?
Do you have to kill the rye cover crop? Could it just be, if it's the annual, could it just be shaded out by the developing corn?
Well, so a lot of people will start with something that kills itself, like a winter annual kind of thing that will only grow in the fall. I mean, we're using winter rye, which is designed, or has been selected to survive the winter and regrow. It's an option to do something that will winter kill. The problem is just getting meaningful biomass production in just the fall. So most of the biomass is put on in the spring, and you get kind of a negligible benefit if you don't get that spring growth as well. Soybeans don't seem to have the same problem with disease following a rye cover crop, and there it seems to be fairly safe to even plant your soybeans into the standing rye and then kill them later or roll them or whatever. So more people leave their rye longer with soybeans than they do with corn. But yeah, and you also plant soybeans later, so you can get a huge rye biomass ahead of soybeans if you want. Deepak?
Your Pythium identification, that was from a culture of the microbiome?
So based on literature and the putative identification of the amplicon sequences, we modified the isolation protocol to be able to get those Pythium strains to grow. Yeah.
But did you do a complementary metabarcode to detect more species?
A complementary
Metabarcode analysis.
Yeah. Yeah. So I'm just kind of picking out highlights. Like we saw in the broader microbiome profile, these taxa that seemed interesting. And so we went and got those taxa out of the site. But there were other things in the profile as well. Yeah. Anybody else on cover crops? Okay, so that was part of a postdoc I did with USDA, and then I moved into a staff scientist position in Peoria with the mycotoxin prevention unit, where I work with other scientists to understand and develop new strategies for controlling head blight. And I was brought in as kind of the microbiome guy to add a new skill to the team, which also includes, plant biologists and fungal geneticists and people with complementary skills. So I had the task of kind of addressing what are all the other microbes that inhabit the same space doing to Fusarium or in this head blight pathosystem, and those types of questions. So that's what I've been thinking about for the last couple of years. Started out with a project for which Ruth helped me, I guess. I don't know if Ruth is here, I don't see her. But she supplied a lot of plant samples from her screening nursery for me, so I'm very grateful for that, and it helped out, made the research that I'm going to present here possible, really. So most of you are likely very familiar with Fusarium head blight because it's been a problem in this area for a long time, but I'll just briefly mention a couple of things about it. So as the name indicates, Fusarium is the pathogen. Around here it's Fusarium graminearum predominantly, and a couple of its close relatives. So it's a residue-borne pathogen that's surviving on stubble and crop residue in the field, and then spores need to disperse from that environment up to the head of the wheat or the barley or the oats. And typically it's believed that most of the infection happens when the plant is actively flowering. And then the issues with the disease are kind of twofold: yield loss because of shrunken seeds and aborted seeds, and you can get like portions of the head where the rachis will die and everything above will get cut off from resources and so on. So you have the yield loss, and then really the bigger economic impact is associated with this class of toxins called the trichothecenes that accumulate in the grain. They're produced by the fungus, and they pose a health safety for people or animals who would consume that grain. So grain is always routinely tested for these toxins, and if it tests high, then you get price discounts, and that's where a lot of the economic impact comes from. And this disease is sufficiently severe that it is really one of the primary determinants of where you can profitably grow small grains like wheat and barley. So for somebody who's interested in microbiomes, is this a disease system that lends itself to imagining or aiming for some type of microbiome-mediated control? Well, I think there are some characteristics of it. So one space that I'm not going to talk about today, but is the survival on residue and the inoculum production on residue. So I have some projects looking at that and how other microbes might reduce the survival or the efficacy of spore production by Fusarium on crop residue. But then also, I think because there's this limited window of susceptibility right around flowering, that we only need control for a short amount of time. If we could get good control for 2 or 3 weeks, that would be huge. So it's hard to imagine, trying to control microbiomes or biology over a whole growing season. But if we have this short window, maybe that's possible. And then also, there's a relatively short period of grain fill in these small grain crops. So the time between flowering and harvest is quite short, and that can limit some options for chemical fungicides that might have minimum pre-harvest intervals that are longer than that grain fill period. So I think it is an appropriate system to consider microbiome-mediated control. But there's this huge issue that I'm still always trying to figure out how to even think about, which is the spatial scale at which microbes are microbial communities are organized. It's like so vastly unlike anything else that we think about that I don't think I ever quite have my head around it properly. So if I'm interested in what's happening in a head of wheat, that seems like a small thing. But relative to the size of a individual spore or a bacterial colony or something, it's equivalent in extent to hundreds of kilometers for us. And so how do we think about that appropriately? I'm not sure if made too much progress on that. I just kind of made an executive decision that I'm going to look at individual seeds that have had the hulls already removed, and that's a simpler kind of defined environment. But if you zoom in, so this is a wheat seed, and at the end here, this is the bearded end of the seed. And if you look at that closely, it's like, that's still, even just that, one individual seed is a really complex habitat. So here's Fusarium graminearum growing on the bearded end of a wheat seed, and it's like, it's almost like a jungle, right? So I need to keep thinking about, am I asking questions at the right scale to understand these microbial processes or not? Is everything we do like continental scale landscape ecology, or can we actually get to communities of interacting organisms? All right, so I just started out by looking in the microbiome profiles of a lot of seeds for some kind of leads, like can we find correlates in the microbiome data for pathogen success in this system or for toxin production in this system. So those seeds supplied by Ruth here from the University of Minnesota from across her screening nursery in 2 different years. So the same cultivar under very heavy management control with the irrigation and everything. And for each of those seeds, we measured 3 different things. The toxin content, quantified the pathogen by qPCR, and then also generated a profile of the bacteria and fungi that inhabited those seeds. So the way we did this, just in terms of the workflow, is just a long extraction of intact seeds, soaking them on, or vortexing them for like 24 hours, 1 seed per extraction, and then taking some of that extract for the toxin, drying down everything else, pulverizing the tissue. DNA extraction goes to both qPCR and microbiome profiles. So I really thought if I try to look at like the whole head, I'm not going to see relationships because it's just, there's such different habitats there, and if I zoomed in on individual seeds, maybe there'd be a clearer story that would emerge. Is that the real time?
Pardon?
Is that the real time? Am I supposed to be done now or at 3:30?
Oh no, 3:30.
Okay.
I mean 3:40, 3:15ish,
Okay.
3:20.
All right. So the first thing that because we did both the pathogen quantification by qPCR and the microbiome profile, I just wanted to see how well do those 2 measurements relate, because it's well known and has been shown many times that microbiome profiles are prone to a lot of biases that skew abundances, and they don't accurately reflect like an absolute quantity. So for this data set, though, it was a reasonable relationship where here's the qPCR measurement. I'm not really showing you the units, and here's the Fusarium relative abundance in the microbiome profile. So you can kind of see that like this plateaus too soon. Once you start getting only Fusarium reads, you can't get higher than that, even though the absolute abundance could continue to increase. But really, that's a reasonable fit. So, I mean, in this case, this was the dominant fungus in the sample, right? Because these plants were inoculated with Fusarium, and most of the fungi that was in there was Fusarium. So it's maybe a best case scenario, but microbiome profiles are not like hopelessly skewed or uselessly biased. I think you can get some reliable information out of them. And this is, again, not surprising information, but the relationship between pathogen, biomass, and toxin content. So the pathogen is making the toxin, so you expect that to be a good relationship. It's not quite as good in the microbiome data as it is in the qPCR, but again, it's a reasonable predictor. So, you get lots of taxa that are present there, and most of them it's kind of like, like go check Wikipedia. What is, you know, I don't even know how to say this one, even though I've read it to myself many times. So it's challenging to know how to pick target organisms out of these lists. This is because I limited my scope to single seeds and stuff. This is actually quite a tame list of taxa, but still many things that I'm not familiar with, have never seen grown in culture, and so on. Interesting among the fungi that, aside from a few basidiomycete yeasts, they're basically all, you know, pathogens or very close relatives of pathogens. So if you're a fungus on a wheat seed, it's like, I don't trust you very much. So for a biocontrol aim, that was a little bit disappointing. I think, you know, you end up, it's all about the strain level characteristics and not just the species ID or something. I'm sure that there are strains of Alternaria that could become useful biocontrol products, but it's challenging to imagine, you know, trying to create and sell and get farmers to apply a product that reminds them of a disease. And for the bacteria, you know, more diversity, fewer things that look like pathogens, but you can see here one of the weaknesses of the particular marker gene that I chose. It's kind of unable to distinguish among members of this whole family, so I get a big chunk of my reads. I can only say, oh, they're Enterobacteriaceae, but I'm not sure we could tie any meaningful function to the family level, so that's another challenge. But so looking across these individual seeds, on average, you find 71 bacterial OTUs, or sequence variants, basically, or bins of sequences, and 19 different fungal phylotypes per individual seed. So compared to soil or something, that's not much. But if you try to plate out, you know, a seed of wheat and see if you can recover 90 different things out of it, that's pretty challenging. So this is really the core strength of microbiome profiles, is access to a bigger picture of what's there that we couldn't see until just 10 or 12 years ago. So among these disease-related variables, we see the expected relationships. I've kind of shown some of them already. So pathogen load is the qPCR, toxin content, and seed weight. So red is a negative correlation and blue is a positive correlation. So seed weight is negatively related to toxin and to each of two different measures of pathogen load. But if we add on now some traits of the microbiomes of these seeds, we can see, again, very consistent relationships where all of the metrics of bacterial or fungal community, diversity, and evenness, these things are all negatively related to the disease severity, used imprecisely, but. And so that's nice. It's a little hard to interpret the fungal side of it since the pathogen is such a dominant component of the fungal community. So yes, of course, there's going to be fewer kinds of fungi where the pathogen is, you know, overgrown the seed. Yes, of course, there's going to be an uneven distribution of taxa because the pathogen has, you know, proliferated massively. So I'm not sure whether we can successfully, you know, extract the pathogen out of that community and then still learn anything new, or if it's just about, yes, this is what happens when one community member dominates an environment totally. But that issue is not there in the bacterial community profile to the same extent. So we still do find, you know, bacterial diversity is higher where toxin content is lower and things like that. And the toxin is not, bacteria are not sensitive to the trichothecenes. Just as a function of their ribosome structure, they don't experience the toxicity. So it's not that they're, you know, being poisoned away. Okay, so another way to kind of show the same relationship. So here are some ordinations where each point here is a seed, a kernel of wheat, and they're arranged in this ordination space according to their either bacterial or fungal community profiles. So seeds that are, or points that are close together in this ordination have similar bacterial profiles or fungal profiles. And we can relate other kind of metadata to these ordinations like this. So these fit significantly. You test that with like a permutation type test that you could read this as you move in ordination space down here, you encounter kernels that have higher toxin content and higher pathogen loads. And in contrast, up here, we have higher seed weight and microbiome diversity and so on. So you'd interpret like a right angle among these vectors here as two variables that were unrelated to each other. And vectors pointing in opposite directions are essentially negatively correlated. So but the point is the microbiome profile fits significantly this other data that was collected from the same seeds. So there's a relationship there. So for instance, we can use things like our diversity indices to model pathogen load, and they will be significant factors in such a model. I don't know, hopefully you can read that, but so if we have pathogen load as a function of really head nested within year and then fungal community evenness and bacterial richness, we can explain half the variation in Fusarium load among these kernels. That's not too bad. As I kind of just did without describing too clearly in the cover crop story, I think what you really want to do is use the microbiome profile to generate something very specific that you could test directly. So in this case, can we drill down into the bacteria that are present there and find particular taxa that may become candidates for biological control or something like that? So these plots are some of the output of a random forest regression of pathogen load based on the bacterial community profile. And you go through this iterative process of kind of leaving out one taxon and seeing how well you can explain pathogen load in its absence compared to when it's included in the model. My understanding of random forest regression is not that strong, but that's sort of the plain language version of it. So if we do that in this data set, we get two different bins of sequences that both look like they belong to Sphingomonas. And the relationship there is negative. So where pathogen load is low, Sphingomonas abundance is high. Where pathogen load is high, Sphingomonas abundance is low. But we don't know whether there's any causal relationship or the sign of that relationship. So does Sphingomonas prevent pathogen success, or does Sphingomonas disappear when the pathogen is successful? We can't untangle those possibilities from this type of a profile. And we need to do that kind of thing if we want to get a management recommendation out of this. Ultimately, we'd like to have less toxin in our grain. We'd like to control Fusarium head blight. I don't think we're going to get there by aiming for maximal bacterial richness or something. I think we're going to need to have specific organisms in mind for our management. So kind of soberingly, if you go back to these ordinations, really the biggest pattern there by far is just the year of sampling. So all the seeds from 2016 are in red and all the seeds from 2017 are in blue. And this is the same cultivar. This is a extremely heavily managed environment, with the mist irrigation and everything. And yet, there's this huge year-to-year effect. So what does that say about our capacity to manage microbiomes? I'm not sure if it's a promising message. So I decided this Sphingomonas thing, I should just go and test this directly because it's the clearest signal in the data, and I should put Sphingomonas on some wheat plants and see how they interact with Fusarium. So, I don't know, presenting negative results is probably as inadvisable as April Fool's jokes, but here are going to be some negative results. So just on a first pass, not optimized, and I know already I made some mistakes, but I put some Sphingomonas out on these plants and put a sprinkler on and inoculated them with Fusarium and is there any protection afforded? So basically we have two different cultivars, Hopewell and Malabar, one without any bacteria added, and this is the Sphingomonas that I used. So we see the cultivar resistance. The Hopewell is more sensitive and the Malabar is moderately resistant. So this is proportion of symptomatic spikelets on the head. So disease progression is faster on the sensitive cultivar. But the yellow versus gray is the treatment effect, and there's no treatment effect. And if we look at the toxin, again, no bacteria added versus adding Sphingomonas to those heads ahead of the pathogen, no reduction in the toxin. And in this analysis, we separated the seeds from the rest of the head tissues. But so in one case, even an increase in toxin in the chaff. So I had this good-looking signal in the microbiome data. I tried it out, and it didn't work. So that's just kind of a cautionary tale. And like I said, I know I could tweak that system to make it a little bit more amenable to control or whatever. But this was just try to put it out under semi-realistic conditions in a field setting and see what happens. So, but I also did some microbiome profiles on those samples, and we could find a few taxa that respond to the addition of the Sphingomonas. So this is among the fungi. There are just a handful of fungal taxa that are either more abundant in control heads or more abundant where the Sphingomonas was added. But conspicuously, Fusarium is not among these taxa that were significantly impacted by that treatment. However, having that data, you can always generate some new hypotheses and find some new patterns. So for instance, if we just compare the fungi that are present in the chaff of this experiment versus in the seeds, there's a number of groups that differ strongly based on that. So Fusarium is able to get into the seed very well. Alternaria gets into the seed very well. But all these other organisms, what is it that makes them unable to get underneath the hull, or why can't they also colonize the seed in the same way that Fusarium can? You could generate, 1,000 interesting questions about that. Okay, so this is kind of what I've been saying, and I just put it up there again as a summary, but my basic thing is microbiome methods are a very useful tool. We've got to be upfront about their limitations. We've got to recognize their biases. But for seeing past cultivation bias, which is also tremendously important, that's a huge plus. Sample throughput, a huge plus. You can do hundreds of samples easily. You don't need to have ahead of time picked your taxon of interest or decided already what the causal organism is in some phenomenon. You can be a little bit broader in your survey. So I'd encourage anybody with a tricky problem that might benefit from a broader survey of the microbial community to try out these methods, or to find somebody who can collaborate with them to do so. And then Linda mentioned already that I'm moving to Canada, so I'm just going to say that clearly, and I'm recruiting for my first graduate student. I've got a project with Delanta Fernando in plant sciences to look at malting microbiomes, and specifically trying to connect Fusarium success within that community and environment to traits at the strain level of Fusarium and to microbiome level characteristics as well. I start there July 1st, so probably the student shouldn't start before me either, but if you know somebody or are an undergraduate, get in touch with me and I'd love to discuss that. So I realized after I had sent this to the office that I forgot my acknowledgement slide. I mentioned a few people along the way, but the cover crop work was with ARS in Ames, so Tom Casper was my agronomist supervisor. He's retired now. Allison Robertson at Iowa State also worked a lot with us. And then this head blight stuff, we have our technical staff in-house that do the sequencing and everything for us, which is a huge resource and a great service. So I'm really indebted to them. And then also my colleague Susan McCormick is a chemist who did all the toxin analysis for me. And Ruth I've mentioned as the source of that material. Thanks.
I think we have time for a couple of questions, and Matt, you can field your own questions.
Okay, anybody? I try to turn the lights on here.
All lights, the top one.
Yeah.
So you saw a shift in the PCO plots for the 2 years. Do you think that the environment, especially the weather conditions, could affect the shift more? And it is better to look at a wider range of time to see if there are more overlaps rather than just 2 years which don't overlap much?
Yeah. So even though it was a managed environment, it's only partially managed, and there's lots of variation that is attributable to the weather in a given year or whatever. Yeah, I don't know whether looking across more years you probably wouldn't see that very clean separation, but you'd build in even more extremes and so on. I'm not sure that a clearer pattern would emerge with more time. Yeah, Linda.
So pennycress is big in Minnesota as
I've heard that,
cover.
yeah.
And, what do you think of pennycress as a cover?
Well, I've never worked with it. I don't know really how it grows, but does it, if it produces enough biomass to be meaningful, I'd be interested in it as a way to preserve your rotation effect in a rotation that includes corn. Having something like pennycress would be great. Although I've heard it shares some pathogens with soybean.
So a little bit of a follow-up question. So you noted that when you used rye, that you ended up with your Pythium.
Yeah.
We had looked at rye a long time ago before you were in the lab, and what we found was that rye was particularly bad for potatoes because it suppressed good streptomyces. And so I wonder if you looked at
Didn't look at
your old
that.
friend.
Yeah, no.
And then, can I ask one third follow-up?
I mean, we did do some bacterial microbiome profiles, and I don't recall streptomyces standing out as being impacted one way or the other.
At first it was the phenotype, not
Yeah.
which again,
Not the
your
density,
microbiome
yeah.
point is that you would miss that,
Yeah.
the functional capacity. But the third thing is, you noted that year had such a big effect on the microbiome, and that might be a cautionary tale. Gosh, can we really manage this? But of course that was absent any management. And inherently, if immigration and everything else, the environment, everything else differs from year to year, and you're not attempting any microbiome management, the variation with year to me isn't that surprising, but does that inherently mean that it's not going to be conducive to managing to some consistency if we think about it, if we actually take into account the community assembly process?
I mean, it wasn't managed for microbiome, and it was managed to promote the success of Fusarium. So to that extent, it was. But it was. I mean, the kinds of management that can be feasibly done were done, and so I feel like that was heavily managed, even though it wasn't deliberately aimed at something like we might aim it at. I think you should still see some signal of I mean, maybe I should have had an unmanaged control in that, but I'm probably missing your question.
No, I mean, I was trying to decide if that was actually a bummer, is that a caution, or is that you know what, we have to think differently,
Yeah.
right? Because when do we expect to have the capacity to manage, and what is it that we need to think about to actually actively manage that microbiome? Is it nutrients on the flower? Is it who we inoculate in what sequence and the timing? I mean, all those other factors that could be really critical to inducing different community assembly processes.
Yeah. Those are good thoughts. Niven?
You mentioned a couple of times about pennycress, and then also Eric and so on, that it has to have enough biomass to be meaningful. Just wondering, when you say that, what do you mean?
So if the benefits you're looking for with cover cropping are, preventing soil erosion, sucking up soil nitrate, those types of things, you don't get there with, half a megagram dry weight per hectare or something. You want 5-6 megagrams per hectare to be meaningful for those ends.
And is that known what you say, or is that it just seems logical? It should be like that?
I think agronomists know that.
Okay.
Yeah.
Thanks a lot.
I'm interested in the experimental design for your microbiome study. So I'm wondering, like in your sample collection, were they from the same cultivar, or it's like you said you are quantifying per seed, like all those parameters? And are you like from one spike you have some seed more resistant, or some seed more susceptible, or like they are a pool looking for a needle in a haystack? They are all put together, and what's the negative control?
Yeah, they are all the same cultivar. So what I asked Ruth for was what she uses to fill in the gaps in her layout. So the same cultivar is dispersed across the whole field, just wherever she needs to fill a gap, and it's her positive control variety. So it's all the same cultivar of wheat. Sometimes there are multiple seeds selected per head, and we were aiming to catch the whole range of seed phenotypes, severity from extremely small and wrinkled seeds up to plump and healthy looking. We wanted to span that whole range on purpose, but it's all within the context of one cultivar. So I didn't show the raw toxin data, but we had a couple of seeds that were like astronomically contaminated with toxin. We're like, that one seed, if it had made it into sampling at any point, it would have made the whole truckload look terrible. But you wouldn't ever harvest that seed in the field because it was so small and shriveled, that it would blow out of the combine and not be collected. But the threshold that we're aiming for is 1 part per million or below, and I had seeds in there that were like 2 parts per thousand. If you pick individual seeds, you can get a broader range of phenotypes than you would in harvested grain. Matt?
You briefly mentioned potentially managing Fusarium on the skull. Did you do any experiments to look into that at all, or have any ideas about?
Well, yeah, I have some stuff ongoing on that that I didn't talk about, but I've started out there too with microbiome profiles on the parathecia to see what are the organisms that can colonize Fusarium parathecia, and then now I've got some target taxa that I'm testing just in a lab assay. It's easier to show that they just reduce the number of parathecia formed, but what I was really hoping for is like something that could attack a parathecia that was formed or forming and eat out its juicy asca sap and whatever. I'm not sure I'm going to get to that, but I have organisms that if they're in the straw too, you get many fewer Fusarium parathecia. And you could imagine applying something like that in combination with any other, with an herbicide or something. It wouldn't take an extra field pass. So if you had some organism that you could just dump in with your herbicide and that was the only cost, I think that might be feasible. But I'm not totally convinced that inoculum is really limiting very often. I think there's usually a surplus of inoculum, and so you'd have to have a really big reduction to get to the point where the inoculum is limiting. Yeah.
I'm not sure I heard that entire question, but did you do any culture-based methods with the Sphingomonas?
Well, that's one of the weaknesses. Is I just picked a Sphingomonas out of the culture collection that was from Illinois, so I could spray it wherever I wanted to, and so I don't know that it had any of the same traits that the Sphingomonas had that were on those seeds. Yeah, it was an easy way to do it.
Just a follow-up with that, did you try a different inoculation method other than spraying the plant?
Well, I have in other experiments. I also have experiments with endophytes that I introduce either to the seed or to the stem lower down from the head or whatever, but none of those in the field yet. I'm just doing that kind of stuff in the growth chamber. But, yeah.
So the two cultivars, Malabar and Hopewell?
Malabar, yeah.
One, do you know the mechanism of resistance for what makes Malabar Is there a significant difference in the microbiomes between two cultivars?
There were, I might even have it here. Nope, I didn't put it in. There were a couple of taxa that were different between the two cultivars. They were just like some of the yeasts. I couldn't see a reason for it really. Those are not the cultivars I typically work with, but one of my colleagues retired and had that experiment or had that field plot set up, and I inherited it. So that I generally have not worked with those cultivars and can't really answer that question. But I would guess that the resistant one has the FHB1, but I'm not positive about that.
Okay, if there are no further questions, Matt will be available for a few minutes to sell his spoon jewelry.